Morning, everybody. Let's do some embryology. It is Monday, week 3 of the spring 2020 quarter. We're still shelter in place, everybody. And yeah, good time to do some embryology. And yep, so we're going to start the show today is fertilization. Remember, we spent a whole lecture talking about formation of the egg. And today we're going to do something with that egg. We're going to fertilize that egg, and we're going to follow it until it becomes a 16-cell creature. And that's called the Morula. So let's get started. Let's meet that ovulated oocyte. Remember, it's pronounced oocyte. You remember that from the last video. So it's released from the ovary during ovulation. We talked about all that stuff. And just like a spermatozoa, it is haploid, 23 chromosomes, or 1M, or 1N. I got a question for you. Process of meiosis. So we talked about polar bodies being formed during meiosis, 1 and 2. Actually, you get one polar body with each. What about a spermatozoa? How many polar bodies do you get formed during that process? Good. You don't get any, right? No polar bodies. Polar bodies are only during oogenesis. All right. So it's a strange cell because it is, again, stuck in meiosis, specifically in metaphase 2 of meiosis. So if you're a sperm and you want to get through and fertilize the oocyte, there are 3 layers you must pass through. So let's talk about those 3 layers. You probably already know those. We talked a lot about the corona radiata. No, we didn't talk that much about it, but we saw how it was formed in Zona Pellucida. And then there is the vitelline membrane. I don't think people are going to call it that, but you never know. It's just the plasma membrane of the oocyte. So corona radiata, a zona pellucida, and the plasma membrane of an oocyte. I mean, ultimately, you got to bind with the nuclear membrane of the oocyte's nucleus as well. All right, here's a picture of this before we've seen. There's the corona radiata. There's the Zona Pellucida, the ZP. Remember the Z? What's the protein? It's made up of ZP3. I kind of gave it away. I was going to ask, well, ZP1, ZP2, ZP3, ZP4. There's other Zona Pellucida proteins as well. Who's the one that binds to the sperm? And what's the name of that receptor? Good, ZP3. ZP3 is kind of sticking off here. That's ZP3, right? Who binds to it? There's the sperm. SED1. SED1's got SED1 fits into that thing nicely. Oh, jeez. This is why I don't draw on the board. SED1. All right, SED1. Those two hook up, and we'll see what happens today. I think we talked about that a little bit before, but. All right. So how does the sperm get through these barriers? Well, it has 2 weapons or 2 processes that it uses. And capacitation we have to start. In fact, that matures the sperm. And the acrosome reaction. So let's start the capacitation story first. So this is considered the final step of sperm maturation. Of course, sperm could be called spermatozoa. Starts when the sperm's plasma membrane contacts the oviduct. Remember, it's swimming up the fallopian tubes looking for that egg. Where does fertilization occur? I know in the fallopian tube, but where? ampulla of the fallopian tube. So just banging into the walls, which is the oviduct, so banging into the oviduct walls, oviduct's like a lumen, some chemicals are released, and we're not 100% sure what they are, so I didn't put those in. But these gene products are released, and it soaks into the sperm and makes the sperm do some crazy stuff. And that's the process of capacitization or capacitation. Capacitation. So what exactly is capacitation? So these chemicals that soak into the sperm, the first thing that happens is it rids itself of cholesterol. So the plasma membrane of the sperm just sheds itself of cholesterol. And normally cholesterol, we haven't talked about that, but cholesterol blocks capacitation from occurring, or blocks, not capacitation, it blocks fertilization. Let me make a note of that. Sorry, 111. Need to fix that. It blocks fertilization from occurring. Can't bind with a lot of protein in there. And then phase 2 occurs. That's the acrosomes lose. They start shedding some of the glycoprotein coat that they have to make it a little bit thinner. Which also blocks capacitation. That doesn't block capacity. Fertilization should be there. That's probably been there a couple of quarters. I'm surprised nobody caught that before. Okay, so once those two things happen, the sperm goes in, or I guess does that make sense? I'm going back to that. Maybe that still makes sense. I mean, that's a capacitated sperm is a hyperactive sperm. It swims like crazy. So I guess you could say that cholesterol blocks that hyperactive phase. So I guess I can leave that in there. But it can't fertilize it, so it really blocks fertilization. So, but a capacitated sperm, it's like the young teenagers nowadays are multitasking like crazy. Ever notice the movies that young people watch nowadays? They're so active. They have always like 2 or 3 stories going within the movie. My wife and I have been watching a lot of movies, and we noticed some of the old movies it makes them kind of hard to watch nowadays. Anyway, I'm digressing. So anyway, so capacitated sperm has a hyperactive phase, and specifically amongst the most important is the tail beats very strongly. And so it's got like an extra drive. If it does find the sperm, it needs that extra power to power in and start getting through these hurdles. The first hurdle that needs to be overcome, of course, is the corona radiata. That's the outermost barrier. Remember we talked about how this developed? It developed from cumulus auriferous cells. And it's 2 or 3 layers thick usually. A lot of that cumulus auriferous is left behind when ovulation occurs, but not the one right surrounding the oocyte comes with. And it's still there today. Not today, but you know what I mean. The space between the cells there's a matrix there, a gel, and that's very rich in hyaluronic acid. The sperm has to get through that stuff. Sperm's not going to go through the cells. It's going to go between the cells. So capacitated, not much challenge here. There's a nice picture from that movie I told you in the very first lecture you should watch. Incredible animation. Not deep enough, though, for us. Capacitated sperm can swim right through that corona radiata, no problem. Now, how do they do it? Because we said they don't go through the cells, but they squeeze between there. But there's that stuff there. How do they get through that hyaluronic acid? Or how do they get through that hyaluronic acid-rich matrix? Well, they have a weapon, and they secrete this hyaluronidase. So when the acrosome releases hyaluronidase, when it contacts the corona radiata, and this powerful enzyme cuts right through the matrix. Now, is that, I see something wrong here. So capacitated sperm is going right through the corona radiata. How acrosome is released, or the acrosome that's the helmet of the sperm, releases hyaluronidase when it comes in contact with the corona radiata. Yes, so that's coming out of the sperm, that's correct. And that enzyme just kind of sprays out, and it can cut right through the matrix with that process. Now we bump up against the zona pellucida looking for our ZP3 receptor. And this guy is a lot stronger. It's harder to get through. There's other receptors, right? We talked about ZP2 and 3 and 4. We don't really talk about them, but the big one is ZP3. So that stands for Zona Pellucida sperm binding protein. So ZP3 is the main receptor for incoming sperm. We know that. So how do you get through there? Well, the first thing that happens is the sperm SED1 binds to ZP3. We've said that. And we've also, on a side note, we've mentioned about mutations. If you have mutations in the gene that make SED1 and ZP3, you can't have a baby. You'll be infertile. So those are very, very important. There are some secondary binding ZP2 could do it. There's another one as well. There's other ways to bind, but if you lose ZP3, your chances of having a baby go way down. So anyway, once you dock, the sperm head docks, something called the acrosome reaction happens, right? You can't get through and fertilize if you're a sperm. You can't get into the oocyte if you can't generate the acrosome reaction. Here we've seen this, I think, 3 times now, but you can see how there's the sperm head and there's the SED1 protein, and it's grabbed this ZP3 protein. GALT is another one, stands for what is it called? Glycos. Oh, there it is right down there. Glycostransferase is another one that can bind, a secondary one, but the important one again is SED3. That GALT also binds to ZP3 proteins as well. All right, so what's the deal with this acrosome reaction? The binding triggers the capacitated acrosome, which is the sperm head or helmet, to release some really important enzymes. The big one is called Acrosin. Acrosin is released, by far the most important. In fact, I cut the other ones out. There are other ones, but this is the big one. That's a chiropractic book, talks about it, and Carlson, which is the best one. So it's a serine protease inhibitor, and Acrosin moves through the zona pellucida and enters the plasma membrane. So it kind of soaks out of the sperm, and we're trying to get through the zona pellucida. It goes right through that, and it hits the plasma membrane. And so what? What does it do? It stimulates the release of some lysosomal enzymes from these cortical granules. And we'll talk more specifically about these after a rabbit hole will go down. But these cortical granules, they release some enzymes that turn off ZP3's ability to bind with other sperm. So before getting through, this kind of closes the door so only one sperm can get through. Okay, so these enzymes cause the zona reaction, and the zona reaction makes sure you only have one sperm to penetrate the oocyte. Okay, and it also, because we've docked as well, these enzymes also cut through. I forgot to put that in here. The enzymes actually help cut through so the sperm can get through the zona pellucida as well. So not only do they have that side effect, I gotta write that slide down. I'll fix that before I post it in Brightspace. But yeah, that's how the sperm gets through the acrosome, by the release of acrosin and some other ones too. So after the sperm penetrates the zona pellucida, it enters a space between the zona pellucida. Here's the oocyte's plasma membrane. There's the zona pellucida. There's the sperm just coming through. This is called the perivitalin space. And once it penetrates the zona pellucida and gets in this space, fertilization has begun. So that's an important concept. When does fertilization occur? Well, it doesn't occur all at once. It's a 24-hour process, but it starts when the sperm head penetrates the Zona Pellucida. Great, easy board question for all medical, chiropractic, physical therapy, all you guys. So make sure you know when fertilization starts, and it starts when you penetrate the Zona Pellucida. All right, so what happens next? So after you get through the Zona Pellucida up here, you find some more receptors and bind to the oocyte's plasma membrane. So you kind of hunker down between these little things that are sticking up. What are these little things? Those are microvilla. It's just like enterocytes have microvilla. There's the plasma membrane of the oocyte still goes around these microvilli. So it's a really the plasma membrane has great volume. So that is still considered part of the plasma membrane. And you can see the kind of an endocytosis process. The sperm, part of the sperm anyway, is swallowed and goes to the inside of the oocyte. But we'll show you something weird about that. So make sure I got everything after swimming through the space. Sperm binds with some receptors on the plasma membrane. It's important to know that the binding occurs in an area that has a lot of these microvilli. The first polar body that was formed, remember that's still hanging around in the oocyte. We haven't completed meiosis 2, so the second polar body hasn't been formed yet. First one's hanging around, and where it hangs around, the area it doesn't have these little microvilli, so the sperm won't bind over the top of that. If the sperm did happen to bind over the top of a polar body, it would be fertilized, and you can't sustain life. There's no plasma, no cytosol in there, and it would be a miscarriage. So, who are the receptors? We didn't say who they bind to. It's not SED1 or ZP3, but there's kind of sorry we asked that question because the binding to the cell membrane is a complicated process. So there's the little, this would be a very hard question. So on the sperm's plasma membrane, it has these binding receptors, atoms 1, 2, and 3. That's not too hard. And this azumo. And these are the 4 receptors that can bind to the oocyte. Who are the receptors on the oocyte? CD9, CD8, and one called Juno, and then integrins. So all of these receptors can bind. They don't all have to bind at the same time. You can bind one of them to one of them. But once you get through that Zona Pellucida, it's a lot easier to get into the center of the nucleus. The hardest thing is getting through that Zona Pellucida. So after binding, so we're going to enter the cytosol, the oocyte's cytosol. After binding to the oocyte plasma membrane, most of the sperm is able to go via endocytosis into the cytosol. But strangely, the sperm's plasma membrane is not allowed in, and we don't know why this is. And same with the mitochondria. They're kind of left as a skeleton on the outside. I'll show you a picture of this in a second. So that's very strange. But nevertheless, the plasma membraneless sperm head and tail, and the tail still works even without its plasma membrane. It's able to swim inside the cell of the oocytes in the cytosol. And now that little tail and sperm head, they're looking for the nucleus because that's where the genetic material, that's where the 23 chromosomes are. And their job is to bind. The sperm head is basically the nucleus that carries the genetic material, right? So we want to bind the sperm head with the nucleus of the oocyte, and we want those two to come together and share genetic material. And that's the next step in this thing. All right, so the presence of the sperm being in the cytosol also triggers some very important stuff going on. Or before I even say that, here's a cartoon of this. So we got of this fertilization process and getting inside the sperm. So we made it through the zona, or we made it through the corona radiata, made it through the Zona Pellucida, and then we made it through the plasma membrane of the oocyte, but for whatever reason, only the sperm's head and tail without the cell membrane is allowed in. So that means this is not going to stay alive very long. It's really got to find the nucleus of the oocyte now and do its job. Why don't more sperm get in? Now, we kind of said the story to this in a second, but that's going to take us down a little rabbit hole. So what happens if more than one sperm does get in? That's called polyspermy, when more than one sperm gets in the oocyte, and that's not a good thing. You get these, it's super, super rare to have someone born like this, but hermaphrodites are something like this, and there's only, I think, 2 of them studied that have ever really been born. So most of the time, if 2 sperm get inside an egg, the egg's not going to make it. But there's a lot of twins, so there's other ways you can have twins. We might as well cover those right now. So there's 2 types of mechanisms for creating twins. My daughter just had twins. So the more rare type is a identical twin or a monozygotic twin. So in this case, the fertilized egg is the problem, or what went wrong. Only one sperm fertilizes an egg. And during cleavage, which we'll start talking about today, it cleaves at the wrong time and creates two separate fertilized eggs, so to speak, and they both grow on their own pathway. So one sperm fertilizes the original egg, and then the fertilized egg is cleaved into 2 pieces, but each of those undergoes cleavage. Cleavage occurs, we'll see when we get there, but fertilized egg is cleaved into 2 pieces, and each of those pieces divides and divides and divides. Normally, cleavage happens within one Zona Pellucida, but it splits. The fertilized egg splits into 2 Zona Pellucidas. It's almost like the egg splits in half, and it's beyond the scope to dig into that any further. But these type of monozygotic twins, or identical twins, they have 100% the same DNA. They'll be the same sex. They'll typically share the same placenta. Now, my new twins, or my new grand twins, those are, of course, fraternal twins, or dizygotic twins. They're 2 different eggs have been fertilized by different sperm. So remember, during ovulation, the right ovary ovulates an egg. Then the next month, the left ovary ovulates the eggs. The ovaries take turns ovulating eggs. But what happens if the right and left ovary ovulate eggs at the same time? Or what happens if the right ovary ovulates 2 eggs at the same time? That usually doesn't happen, but it could happen. And those will be 2 different eggs, and 2 different sperm fertilize those eggs, and you got yourself a fraternal twins. So these twins don't, because they're different sperm and different eggs, they share only about 50% of their genetic material. So they're similar, but not any more than a regular brother. Another brother who wasn't a twin. They share about 50% of their genes as well. So they can be different sexes. And in fact, my twins, we have 1 boy and 1 girl. All right, so here's a little recap of that. So fraternal twins, for whatever reason, we had the ovulation of 2 eggs, and both of those eggs got fertilized. And there we have Joe and Pam. Identical twins is we had 1 fertilized egg, and then something goes a little cuckoo with cleavage, and you cleave into 2 different, or even 3 different, or even 4 different in some cases. You cleave into 2 complete kind of cell lines here. All right, those are identical twins. 100% of their DNA is the same. Polyspermy is, I want to say, I don't know if I should say luckily, but it doesn't happen very often. People aren't born once polyspermy occurs. But what it is, polyspermy, just like the word says, 2 sperm have gotten into and fertilized one egg. And that creates not a haploid zygote, not a diploid zygote, but a triploid zygote, because there's 3 sets of chromosomes in the mix when this happens. There's one from the egg, and then there's one from each of the sperm. So we got 3 sets of genetic material. And if they survive, they're called semi-identical twins. So let's look at these survivors, semi-identical twins. They're also called sesquizygotic twins. So these guys are weird because normally, let's back up. So if I take a cheek, I scrape your cheek with a toothpick, and we look at your cells and we analyze those, and we can learn that you have a certain amount of DNA from mom and a certain amount of DNA from dad. If I go down to your big toe and take some cells down there, it'll have that same proportion. Same with the fraternal twins. You're always the same mix because you started from one cell. But not these sesquizygotic twins. They have different amounts of genetic material in different parts of the body. So it's very strange. So all the body cells have different proportions of maternal and paternal DNA. So really weird. Usually, as I said, results in termination of the embryo or fetus, doesn't make it. A couple have been studied. There's an Australian semi-identical twin, and based on their genetic testing, 100% of mom's DNA is in all of the cells, but not so with the dads. The two dads both contributed different amounts of DNA to the cell. So in skin cells, 70% of the DNA was from a dad, but 58 in the gonadal cells. And you go to different tissues and you get different mixes. So it's very, very strange setup. It's thought to happen in about 1% of pregnancy, so that's common. But most of the fetuses are the zygotes are lost. Can't survive like that. In fact, to date, last check in 2019, I checked the research. There's only 2 sets of polyspermy twins or semi-identical twins have ever been born and studied. Both of them were hermaphrodites because they end up with a breast on one side and they end up with a male breast on the other. Maybe they'll have a vagina and a penis. They're kind of a mixture of sexes because of the double sperm material. So many of these children those are called hermaphrodites when you have human male and female parts on the same body. So this is Hermaphrodites, the Greek goddess. It's super, super incredibly rare. Like I said, there's only 2 that have been studied. Although there's thought to be a lot more, and people you're obviously not going to let your kids be studied by science. How do we prevent polyspermy? Now we're getting back to that. So we need to talk about fast block and slow block. And I got that one slide out of place because we kind of talked about it a little bit. But let's talk about fast block polyspermy. So within seconds after fusion of the sperm and the oocyte's plasma membranes, the entire oocyte plasma membrane depolarizes like crazy. So a massive depolarization occurs. And this is one mechanism. If we get into we didn't talk about this, but you need to be like a normal membrane, about negative 70 millivolts, in order for a sperm to be taken through by endocytosis. If you're down to negative 40, or if you're positive, other sperm can't get through. So that's called fast block polyspermy, and that occurs when the winner, the champion sperm binds to the oocyte's plasma membrane. It prevents other spermatozoa from coming in. Right? Then we have slow block polyspermy. So slow block polyspermy occurs via calcium as the star of the show here. So immediately after the sperm enters the cytosol, it releases something called phospholipase C zeta, and that comes out of the sperm. So it causes the oocyte endoplasmic reticulum to start cranking out waves of of calcium ion just sweeping through the cytosol. And that calcium ion does a few things. It stimulates the restart of the oocyte's second meiotic division. Remember, the oocyte was stuck in metaphase 2 of meiosis. So that wave of calcium stimulates that guy to get going. And it does. The oocyte finishes out mitosis 2, and it creates a new polar body. And now we have 2 polar bodies in that cell. It also causes, here's the cortical granules. So it causes these cortical granules to fuse with the oocyte's plasma membrane, and they release their stuff into the parivitylene space, right? And that was the space between the Zona Pellucida and plasma membrane. This causes a decondensation of the nucleus, so I'll tell you right now, so this also kind of with metaphase 2, but after the oocyte's nucleus, after it goes through metaphase, or after it goes through meiosis, it usually stops. You think it would stop and not do anything more because we've completed meiosis. But it actually goes into interphase, which is really strange. Because, what is that, a meiosis 3 we're starting? So it's a really strange type of thing. But the chromosomes, they get ready for interphase. They shrink, right? The meiosis and mitosis, the chromosomes are very thick, and we can see them. But most of the time they spend in that interphase, and they're all unwound, and they replicated in that process. But it causes decondensation, makes them get ready for interphase. What's the deal with cortical granules? So they float around in the cytosol, minding their own business. But when they're stimulated to do so, they are going to release 2 types of molecules into the parivitylene space, polysaccharides and proteolytic enzymes. We'll just leave it at that. Polysaccharides are what cause the Zona Pellucida to stiffen and swell. So this is yet another mechanism that prevents polyspermy, or more sperm from getting in there. That's actually called the Zona reaction. Okay, Zona reaction. And that's part of slow block polyspermy. So polysaccharides cause the Zona Pellucida, and we won't name them all, but it makes them get stiff, and it's really hard for other sperm to get in. That's the Zona reaction. What about the proteolytic enzymes? They destroy the remaining ZP3 receptors. So that's like the third one, or fourth one we've talked about, that block other sperm from getting in. So if you destroy the ZP3 receptors, you can't bind, obviously. So that's yet another mechanism. Right? So all of these mechanisms we talked about, including the membrane depolarization, they all inhibit polyspermy. All right, so now if you're, again, here's the amoeba sisters. Go look at the amoeba sisters, meiosis and mitosis. It would behoove you. I think Dr. Doe's going to hit you with that as well. You're going to get hit by mitosis in several classes. You cannot escape the cell cycle. So go refresh on that. I'll assume you know that for the most part. But as I was saying, remember interphases. The chromosomes are wild and crazy and super small. You can't see them anymore. They're so small. They thicken up in mitosis and meiosis so you can see them. Here are the phases again if you want a quick review. And remember, we were stuck in metaphase 2, but once the sperm got into the cytosol, it started up again. And we went through anaphase 2, telophase 2 cytokinesis, and we got ourselves 2 new creatures from one creature. We got another, but this is the polar body, right? But now we have a mature nucleus, whereas this part we didn't. All right, so make sure you know that stuff. So after penetration of the plasma membrane, the oocyte resumes meiosis. We said that. Waves of calcium stimulated that, we said. Meiosis 2 kicks into gear, and then meiosis is completed. the sperm is also looking for the nucleus of the oocyte at this time. What else happens after penetration? So we create a mature oocyte, right? So it's going to be haploid still. It's going to have 23 chromosomes, but it's mature. There'll be a second polar body that was formed. It didn't kind of double like it does with mitosis. Anyway, fertilized oocyte gets a new name. It's now called the definitive oocyte. Moore calls it kind of a weird name, the ootid. The other books don't use that. So ootid is kind of weird, but Moore is one of the board books, so I had to throw it in there. So, but that's a fertilized, or at least it started the process of fertilization. It hasn't completed it yet. All right, I guess I thought I took these out, but I did leave them in. So wave of calcium created. We went through that already. So there's the second polar body being formed, though, because we're forming new cells, but this one steals all the cytosol. Okay, so definitive oocyte is a really weird creature, right? Up until it actually binds, until you form a zygote, because it's weird because you have two nuclei in one cell. You have one from the sperm head, has a nuclei that's in there. That's fully mature, right? That's already went through meiosis too. And now you have the oocytes nuclei, which is now are almost going to be mature. I mean, as we speak, it's maturing, going through meiosis too. So basically, we're going to have two mature nuclei, but they're both 1N. So that's a really weird cell. So our goal is to make one normal body cell or somatic cell that has 46 chromosomes, that's 2N, it's diploid. And that's the next task. So jumping the gun. So this is very strange. So interestingly, as already said, the male and the female nuclei, which are now have went through meiosis 2, there's no meiosis 3, right? They're done. But for whatever reason, they start replicating their genetic material again by themselves. They're not bound together yet. They're separated, which is really, it's almost like they're starting a meiosis 3, I guess, because they're 1N. Mitosis only occurs in cells that are 2N, have 46 chromosomes. So it's really strange. But yeah, so they're duplicating the chromatids. The centromeres are still 1, though. Those never duplicate, right? So what they're doing is they're getting ready to join each other and morph together. And they actually make it into prophase. So they start thickening and become visible. And that's the time when they're going to come together, like the great Beatles album here. So let's change this weird ootid into a normal cell. That's what we're going to do by coming together. And we pull these nuclei together and let their genetic material mix. So there's the sperm. You can see we're in prophase because we can see the thickened genetic material now. But these are still only 1N. But if we combine these together, we have a normal cell. But this cell right here is pretty darn weird, you'd have to agree. What cell has 2 nuclei in it? So everything I've already said. Meiotic interphase is already started. Once the chromosome genetic material thickens to this point, these get a new name, and they're not called the nucleus anymore. They're called the pronuclei. That means that they've went through interphase, and they've went through interphase. They're getting into prophase now because they're thickening, and that's called pronuclei. And that means they're almost ready to bind together. That sperm tail is just about degenerated away. You can barely see it. And coming together, here's a beautiful shot, right, of a definitive mature oocyte, which is weird, right, because there's those 2 nuclei. But it's not going to be like this for long. This membrane is going to dissolve. This is the male, this is the female. This membrane will dissolve, and the genetic material, the chromosomes will all mixed together. Remember, each chromosome has only one centromere at this time. All right, so here's one centromere, but each one through interphase, they've duplicated the chromatid, right? But that's still called one chromosome. So that is what's going on. Each one of these cells has 23 of these chromosomes, even though they have 4 chromatid. So that's always a weird thing. Remember, after they're pulled apart, chromosomes look like this. They have a centromere with only one chromatid. That's still, this count here, each one of these is one DNA, or one chromosome, right? These are each one chromosome. The only difference is these are ready for mitosis or meiosis 1, because they're about to get ripped apart, but that doesn't change the chromosome count. Chromosome count is 1 here. Chromosome count is 1 here. Okay, go back to the amoeba sisters if you don't get that. All right, so let's get these guys mixed together. And that's exactly what happens in this nice picture. We can see the two nuclei have bound together. The membrane where they bind dissolves, and the genetic material, the chromosomes, mix together. All these double 2 chromatin armed chromosomes mix together. And yeah, at this point, fertilization is over. We've completed the process of fertilization, and this is no longer called that definitive oocyte. It's called a zygote. Many people believe this is the beginning of life right here, right? All the chromosomes are still in prophase, but they're going to line up. They're going to head into mitosis, metaphase of mitosis, M for middle, as the amoeba sisters will teach you, and they're going to line up. And anaphase, away, A for away. They're going to pull apart an anaphase, and we're going to split this. We're going to split this cell through a process of cleavage into two cells through cleavage, and that's where we're going right now. So meet the zygote. Well, I already introduced you, but the formation of the zygote is the process of fertilization. We've just created a new cell, which is now a regular cell. It's diploid, 2N, 46 chromosomes. Yeah, it's a real cell. And how many chromatin does it have in this state right here? Ooh, 46? No, 92. There's actually 92 chromatid. There's still 46. But there's that, when this splits in half, it'll go back to 46 chromatid. Make sure you know stuff like that. I'm assuming you know that. I'm not sure who teaches genetics. I'm not sure where that's supposed to be. But it's definitely on boards. Medical boards, it's genetics. Medical school, we have genetics class. We don't have a dedicated genetics class in chiropractic, which I don't think is a great idea. And why that is, I don't know, because genetics is super important. Anyway, I'm digressing again. So once the zygote is made, we have the first stage of a human being. And the process of embryogenesis, now that fertilization is complete, the process of embryogenesis has begun. So that's a key word to know, embryogenesis. Some authors call it organogenesis, which I don't like it. Embryogenesis is better, but be familiar with that word. And so embryogenesis is from ground zero, right after fertilization occurs, up through the 8th week. And once you get into the 9th week, that's called the fetal period. So the fetus doesn't come around until week 9. So up until then, it's always a zygote or an embryo, which we're going to form here in a second. Good slide to make questions from. What are the results of fertilization? Well, we stimulated the female gamete to complete its second meiotic division, so we successfully got that fixed. It was stuck in metaphase 2. We've restored, by the genetic material of the male and female, the pronuclei morphing together. We have fixed the weird cell, and made it a normal single-cell creature, single-cell zygote with 2N, 46 chromosomes, right? 92 chromatid. Let's see, and then the crossover already occurred. Crossover occurred way back in meiosis 1, so that was a female thing. That crossover gave us a new, mixed all the chromosomes up between mom and dad. And the zygote is created, yep. Sex is determined, right? The male determines sex. We probably know that in junior high by now, but the male is the one that determines the sex because the oocyte only has an X chromosome in it. And that's its genetic, or that's its sex chromosome. It has to be X. But the male can have a X or a Y chromosome in it. Therefore, when that genetic material mixes, you can have an XX. If the male was a Y chromosome, you have yourself a female. If the male sperm was actually a Y chromosome, it's like a male sperm or a female-ish male sperm. I don't know if that made sense, but I think you know what I mean. If it was, because they only carry one sex cell, right? The sperm are haploid, so it can only carry a X or a Y. So a Y sperm will make a male, and X will make a female. All right, so got our zygote going on, and that's not going to last long. We're going to create an embryo really quickly. So let's create a two-celled zygote, which is called an embryo. So currently we have a single cell, diploid cell, we know that. Why did I put these in here? I don't know. We'll have to make a human with 37 trillion cells. So we got a lot of mitosis to do. How are we going to do that? Cleavage, cleavage, cleavage. We are still going to split the cells in half, but this time not by meiosis, by mitosis. And then we're going to repeat that process over and over and over again. So here's where we're at in our single cell zygote. This might as well be the zygote right here, and there's its nucleus, and it has 46 of these 92 chromatin, and yeah, this is where when we came together. So now the whole cell is mixed. Now the first thing that happens together is one unit. We go through my metaphase, M for line up. Anaphase is away, as the amoeba sisters say. I really like that. And they pull away. It'll teach you, how that works. And then telophase and cytokinesis was left out, but the cytokinesis that walls off. And now we got two daughter cells. In embryology, we don't call them daughter cells. We call them blastomeres. They're still daughter cells. I don't know why they do that, but they're called blastomeres. So great. And that process is going to happen. Now, each of these is going to go through the cell cycle again. Now we're going to have 4 over here. And then each of those is going to happen. Then we're going to have 16. They're going to split, and they'll go through mitosis again and again and again. All right. Now, let's see. What else did I want to tell you? So these are still encased by a single Zona Pellucida, right? So this is happening inside mom's, that egg, right? So there's what's not called a zygote. We can still call it a zygote. But this splitting still has the Zona Pellucida around it. And maybe the corona radiata still hanging around. That hangs around for a few days. So this is going on inside the egg. Okay? It's an important concept. All right. So meet the two-cell. Well, we just met the two-cell zygote without the Zona Pellucida around it. But once the zygote has cleaved into a creature with 2 daughter cells in it, you'd get a name change again. Now it's going to be called an embryo or concepsis. Some people, other experts, think this is where life begins. Our board books say it's when the genetic material mixes, so that's what we're going to use. But you can read about that and argue about that all you want, but I don't think it makes that much difference because it happens so quickly. But anyway, so now we have an embryo or a conceptus. You can still call it a zygote if you can't remember. I mean, all the way up through the embryonic period, you can call it a zygote if you want. And then remember, daughter cells are called blastomeres. Each blastomere has a normal body cell count, 2 and 46 chromosomes. All right, there's a really cool shot of a human embryo. Now, would you remember, is that fair to call that an embryo? Yep, because once you get 2 cells, you're an embryo. Okay, and there's the Zona Pellucida correctly. This is the Zona Pellucida. You can still see one of the polar bodies, the other polar body's right there. It's got 2 polar bodies degenerated away. There's a Zona Pellucida, or there's a corona radiata also around this, but it didn't show up. Right, so each blastomere is going to repeat. It's going to go through mitosis, interphase, cytokinesis, and then we're going to have a 4-celled little zygote, if you will, or embryo, whichever you want to call it. And this embryo is getting bigger. Each time these cells are dividing, for a while, it's going to get bigger and bigger and bigger. Note the Zona Pellucida right here. That's the Zona Pellucida. Right? Blastomeres are everything I said. They're getting bigger at first. They're not always in sync either. It doesn't divide perfectly. Like here's an example of a 9-cell embryo. So one of the blastomeres divided a little quicker than the other one. That's fine, and that's the way it goes. But this thing is getting bigger and bigger until something happens. So around the 8-cell stage, that the party's over. The zygote, the embryo, it's not going to get any bigger. The division still has to occur, right? We still got 37 trillion creature to make. But at this point, something called compaction happens. And the genes, some of the genes inside the nuclei of all these blastomeres, they start making their first CAMs, or cell adhesion molecules. And the first one made is tight junctions. And I'm sure the other ones are made pretty quick as well. But these start kind of pulling the cells together and making them clump. So the zygote or the embryo will no longer grow in size after about the 8-cell stage until we get into the wall of the endometrium. They're stuck at the side. Yet every division, the blastomeres get smaller and smaller and smaller in order to keep this size going on. Okay, then we come to the morula. The early embryologists thought this looked like the zygote embryo looked like a mulberry. And I don't know, it looks kind of too long to me. Maybe a blackberry would have been better. All right, those are a little bit shorter at least, but anyway, that name stuck. So about day 4 now, past fertilization, the embryo enters a new stage, and it can be called a morula. You could still call it an embryo or a zygote if you wanted, but more properly, it's called a morula at this point, so you get an idea of how big it is. Now, the authors are all over the place on this. Some authors say that a morula doesn't occur until the 32, one more division, 32-cell stage. Other authors, I think, wisely say anywhere between 16 to 32 cells, you got yourself a morula. That's probably the best way to go. Right? But because Langman is a board book, we're going to go with 16 blastomeres equals a morula. All my students got that? 16 blastomeres equal morula. If medical students, check what books you guys are using for boards and go with that. Same with physical therapy and acupuncture. Whoever's watching this, make sure you check with your local boards, because I'm gearing this. I teach chiropractors, so young pre-chiropractors. And so I'm geared to teach by the chiropractic board books. It's a really nice electron microscopic view of a morula. And yeah, compaction's still going on, and I think, yeah, we're about done. I don't think we're going to go much further. The genetics is the same. Each blastomere in the morula is still diploid, 46 chromosomes, 23 pairs. How many chromatids? Well, it depends, because they're going through mitosis, so it depends at what stage in the cell cycle they are, or how many chromatids there are. Okay, and you can break it down further. There's actually 44 autosomes in every one of those cells, and 2 sex chromosomes, an X and X or an X and a Y, which gives it the sex. All right, cleavage time points. I think this is the last slide. Larson, I really like Larson. That's a great book. Anyway, cleavage time points. So fertilization occurs, that starts the clock counting. So the first 24 hours, we get 2 cleavage occurs. The first cleavage occurs, and we get 2 daughter cells. The embryo is created. Day 2, they divide again, and we have 4 blastomere creatures, getting bigger too. Day 3, all of these divide again. On average, you get an 8 blastomere little zygote or embryo. Day 4, you get to reach the 16 cell stage, and for us, we're going to say the morula is born. Right, and we'll see what happens to the morula next week. Aren't you excited? It's going to drop into the uterus martini glass, and it's going to bind to the endometrium, and oh, how exciting. All right, so we will see you guys next week. Or actually, I'll see you in spinal anatomy, but I'll see the rest of people who are watching this next week.